Okay, so welcome back to this next video in which we are discussing the detection of protein palmitoylation with tritiumated palmitic acid. Okay, so we've discussed that we have got these cells which have protein A in, which is the protein we're trying to decide whether it has been palmitoylated or not. What we now want to do is lyse these cells open and find out whether protein A is palmitoylated or not. Okay, so the first process that we now need to discuss is cell lysis. Okay, right. So, the process of lysis is going to be done in a cold lysis buffer. Okay, so you are going to basically lyse the cells with a cold lysis buffer. And let me show you what is within this cold lysis buffer. Okay, so firstly it contains a bunch of familiar faces. Okay, so firstly, again, it's very similar to the STE fluid. It contains 150 millimolar sodium chloride. So one of the key components is sodium chloride. Okay, next it also contains tris. So it contains 50 millimolar of tris hydroxymethyl aminomethane. Okay, often just abbreviated to tris. And then also EDTA is present again. It's been upped as well to 2 millimolar of ethylene diamine tetraacetic acid, EDTA. Okay, right. So now some new faces. So in this cold lysis buffer you're going to have other things as well. So you're also going to have 5 millimolar sodium fluoride. Okay, so sodium ions ionically bound to fluoride anions. Okay. Then you're also going to have two other molecules which I want to discuss their structure with you. Okay, so you're going to have 0.5%, okay, sodium deoxycholate. So let me explain what sodium deoxycholate is. So firstly, I need to explain what cholic acid is. I then need to explain what deoxycholic acid is, and then sodium deoxycholate will be the sodium it will be sodium bound to the deoxycholate anion, basically. Okay, so let's start with what cholic acid is. So cholic acid is a steroid molecule. Okay, so let me start by drawing the steroid structure. And more specifically, it's actually a sterol, so it's going to have an alcohol group of this first ring of the steroid structure. Okay, so here is the typical steroid structure, which contains these 4 iconic rings of carbon. And I'm drawing skeletal structures now, because if you try drawing molecular structures for this, it just becomes an eyesore. Okay, so remember, in skeletal structures you don't show carbon atoms, they are implicitly shown by corners, and you don't show hydrogen atoms coming off carbon atoms. Okay, so I said it was a sterol, so we're going to need an alcohol group coming off this structure here. Okay, and we're also going to have methyl groups coming off here and also here, and then we're going to have 2 alcohol groups, one here, well, 2 more alcohol groups, one here and one here. Okay, now to complete cholic acid, we need something that actually is acidic, because at the moment we've just got a bunch of alcohol groups and a huge steroid structure. So up here, you will then have a acidic group, basically. So you have a 1, 2, 3, 4, 5 carbon carboxylic acid group sticking off the side. Okay, so here is the carboxylic acid group there. Okay, and this is the structure of cholic acid. Now, if you want to convert this into deoxycholic acid, all you do is remove this alcohol group here. So if you take that alcohol group off and replace it with a hydrogen atom, then the structure which you then get is called deoxycholic acid. Okay, now if you want to create sodium deoxycholate, then what you would do is you take the proton off this oxygen of the alcohol group of the carboxylic acid group. Okay, so you take that proton off. So remember, this is a hydrogen atom, so it will usually be protium. So it will consist of just a proton and an electron. The electron will be involved in the covalent bond to the oxygen, okay? So you just remove the proton, you leave the electron on the oxygen, okay? So what that will create is the oxygen will then have gained an electron and therefore will have a negative charge. That then is the deoxycholate anion, and when you bind that to a sodium cation, that is then called sodium deoxycholate. Okay, so this will be sodium deoxycholate. Okay, right. So, next up then, sodium deoxycholate is one of the key members of the cold lysis buffer, but there is one other very important member, which is a detergent molecule. Okay, so we're also going to have 1% of a molecule called MP40, and MP40 is short for nonidate P40. Okay, so nonidate P40, and this is usually just abbreviated to MP40. This is a detergent molecule, as you'll see in a moment. So basically, nonidate P40 is going to have a very long hydrophobic portion, but it's also going to have a very, a terminal polar group. So that's why it's going to be a detergent, because it will have a polar group which can interact with water, and then hydrophobic groups which will interact very well with fats. Okay, so let me now show you the structure of nonidate P40. Okay, so we start off then with a benzene ring here. Okay, and then coming off the benzene ring, you then have a 9-carbon alkane group that is fully saturated. Okay, so this will be 9 carbons, where all of the carbons are fully saturated with hydrogens. Okay, so I'm just going to abbreviate this to C9, and then how many hydrogens are we going to have coming off it? Well, every carbon, apart from the very, very last one, will have 2 hydrogens coming off it. So we'll have 8 lots, therefore, of 2 hydrogens. That takes us up to 16. But the final carbon, right on the end, will have 3 bonds available, because it doesn't have 2 bonds to carbons, it only has 1. Okay, so it will have 3 bonds, therefore, to hydrogen. So that will take our hydrogen count up to 19. So C9H19 over there. Okay, and then we have a repeating group. So basically you have an oxygen molecule here, and then an ethylene group. And you repeat that over and over again. Okay, so this can be repeated over and over again. So this will now then be an oxygen here, and then an ethylene group, an oxygen, and then an ethylene group, an oxygen, and then an ethylene group. And you can have that repeating any positive integer number of times. And then finally on the end you have an alcohol group, which is the polar group. Okay, so this molecule then is a detergent, and this will help to dissolve the phospholipid bilayers. Okay, right, so that's nonidate P40. Right, so this now concludes our discussion of the cold lysis buffer. So that's all of the things that you'll have within this cold lysis buffer. What this will do is it will lyse the cells, so all of the membranes will break down and everything will just be released into a gloop. What you will then do is take this gloop and you will centrifuge it. Okay, so we've now got our lysed cells, and somewhere within this gloop that we've ended up with, there will be, our protein A, which is palmitoylated. Now what we're going to do is centrifuge this test tube, which contains our lysed cells. Okay, so we're going to put it in a machine known as an ultracentrifuge, which will basically just twist this tube round and round and round and round and round and round, spin it round and round. Of course, you won't spin it like, you won't spin it like that, because all of the stuff will fly out. Well, all you're going to do is put some lid on the top there, and then spin it round and round and round and round, rotationally, basically. So, well, that's a useless word, but basically, if this is the axis of rotation, basically, so the axis along the test tube is going to be the axis of rotation. We're going to spin it around like that. Okay, so this is an ultracentrifuge that we're going to use to do this, and it will basically spin this test tube round and round and round, and this will basically get out all of the large structures, basically. Okay, so most of the gloop will now come down and form a great big pellet, basically. And what will be left in this fluid on the top after you've ultracentrifuged the solution is you'll have your proteins in this fluid on the top. Okay, so protein A, which is palmitoylated, will be in this fluid on the top, and the fluid on the top is then called the supernatant. Okay, so what we're going to do is take the supernatant, and this will contain a whole bunch of free proteins. Now, we somehow want to extract protein A specifically out, and not take other proteins with us, basically. And we're going to do this. We're going to separate protein A from the rest of the proteins using a technique known as immuno precipitation. Okay, precipitation. That's a long word. Right. Okay, so what does immunoprecipitation involve? Well, basically, it's exactly this. It's a technique for separating a mixture of proteins apart. Okay, so we just want protein A, and remember it's palmitoylated. Okay, so here is the protein A that's palmitoylated, and I'll colour in these palmitoyl groups in the radioactive yellow again. And it's in this supernatant, or this fluid that we've now extracted from that test tube over there, with a whole bunch of other proteins. So let's say this is protein B, over here is protein C. So how are we going to separate it out? Well, basically, what we're going to do is get tiny little beads. Okay, and these are really, really tiny little beads. You can't see these. These are minute. And basically, you're going to attach to these beads antibodies that are directed against protein A. Okay, so this is the immuno in immunoprecipitation. We're using antibodies, and therefore it's an immunoprecipitation reaction. We're going to use antibodies, that's the immuno, to precipitate out protein A. Okay, so here is our antibody molecule, and this will bind to a certain portion of protein A, and the little domain of protein A which this antibody molecule binds to is known as the epitope of protein A. So this bead is covered in this antibody which binds to a certain epitope of protein A. Okay, so here comes along protein A, and it will bind there. And of course you'll want to make sure that it's not binding to somewhere where the palmitoyl groups will affect its binding. Okay, so what you will therefore do is add in these beads which are covered in antibodies against protein A, and what will then happen is protein A, but not all the other proteins in the solution, will then bind to the antibody on the beads. Now what you do is you take this solution with the beads and the antibodies and the protein A attached to it and all the other proteins which haven't bound to the beads, and you centrifuge it again. So you spin it round and round again, and what will happen is the beads will drop to the bottom and form this pellet here. So this time it's the beads that will drop to the bottom and form the pellet. And of course they will drag with them the protein A, which is attached to the antibody molecules which are attached to the beads. Okay, whereas protein B and protein C, of course, they will remain in the supernatant just as before. Okay, so what's changed for protein A is that it's now attached to these beads, and that's the reason it's precipitating out, which just means forming a solid, basically. Okay, so you precipitate out protein A, but not any of the other proteins through this mechanism, which is known as immunoprecipitation. So this technique of separating protein A from the other proteins within that solution by antibodies attached to beads is called immunoprecipitation. Then what you will do is you'll take this pellet out, okay, and you'll examine the pellet for radioactivity. So if you can find a radioactive signal from the pellet, then you know that protein A has got these palmitic acid molecules bound to it, which are radioactively labelled, and therefore protein A must have been palmitoylated. Okay, now let's think what would have happened if protein A had not been palmitoylated. Well, palmitoyl groups would not have been stuck onto protein A. So, when we do this immunoprecipitation, protein A will still bind to the anti-protein A antibody, and therefore will still be precipitated out in this centrifugation process. But when you direct your Geiger counter at that pellet, you will not find any radioactive signal, basically. And that's how you can determine whether the protein A has been palmitoylated or not.